Good morning, everyone. Welcome to the today's Association for Fungal Biologists and MicroIndia MicroTalk 3. Today we have our speaker, Professor Hesham Ali El Enshasy. He'll be talking on fungal morphology in biotechnology industries, taming wild microbes. Before we start, I'll just like to briefly introduce about the AFP. The Association of Fungal Biologists is a scientific body constituted with a broad interest in advancement of mycology and caters to the needs of the mycologists worldwide. It's a very recently established association. The association was formed with a view of bringing together the mycologists with the broad objectives of promoting and disseminating knowledge on different facets of mycology from wide perspective. The major emphasis has been laid on the application of mycology to harness the biotechnical potential of microfungi and macrofungi for human welfare. It also foster collaboration, exchange of ideas, and explore innovative approaches for studying and harnessing the potential of fungi. And definitely, it helps in the advancement of understanding of fungi and their significance in addressing the global challenges, fostering sustainable practices, and promoting our overall well-being. So this has been a very nice platform for the networking opportunities is there, and it is also helping to disseminate the cutting edge research and inspiring the participants for further contribution in the field of mycology. MicroIndia, to tell you about the MicroIndia, is a journal of Indian fungi, which is a premier open access peer-reviewed online journal dedicated to the field of mycology. So it's an open access thing. There is no charges. It guarantees a rapid quality-centric review-based process. It has various global specialists on the board. And at MicroIndia, we promote a broad range of fungal research topics, including fungal biology, biochemistry, pathology, ecology, and genetics. But we are particularly interested in studies that reveal the multifaceted interactions of the fungi with diverse ecosystems and their relationship with various organisms and their influence on human health, disease, industries, and agriculture. To introduce our speaker, Dr. Hesham El Enshasy, the current director of Institute of Bioproduct Development and professor in Bioprocess Engineering, Faculty of Chemical and Engineering, Energy Engineering, University Technology Malaysia, UTM. Since 2020, Dr. Hesham is recognized as one of the top 22% researchers in the world in the field of biotechnology, energy, and enabling technologies, according to Stanford University Research Study of both career and single year. He is also visiting an adjunct professor in Shizuoka University, Japan, and adjunct professor to Universitas Negeri, Jakarta. Dr. Hesham is also serving as a board member and biotechnology consultant for strategic planning, bioproduct development, facility design, and the three T's, that is the technology transfer, implementation, and the development. Before joining UTM in 2008, Dr. Hesham was the Assistant Director of City for Scientific Research for Research Affairs, Alexandria, Egypt. He did his earlier he did his two postdocs from GBF Germany and Ohio State University. Dr. Hesham was also visiting a guest professor and honor guest professor to many of the world-class universities in USA, Germany, Sweden, Japan, Australia, Saudi Arabia, and Algeria. He has more than 60 industrial projects and 400 publications in different niches of industrial biotechnology and serves as editor and associate editor in many international journals. Dr. Hesham has received the UTM Award for the Best Cooperation in Industry in 2018 and 2022 at the university level, and Best Cooperation with Industry and Faculty level at 2019-2020. And Bioeconomy Malaysia and National Award for 2 times, that is 2012 and 2017. With this, we welcome you, Dr. Hesham. It will be a pleasure to listening to you. We welcome you for the talk, sir. Please.
Rohit, let me interrupt. Professor Deshmukh, could you just speak for a minute, please?
Yeah.
I think Rohit has already given the idea about AFB, so you can go ahead with that.
Okay, thank you. Yeah.
Dr. Shah, can we begin?
Yes, please. Yes, please.
Yeah. So please go ahead with your talk, sir. Thank you.
Okay. Thank you very much for Michael India. It's a great pleasure and honor for being with you today, with my friends, Prof. Sonil, other colleagues, other professors and doctors in this platform. And usually it's a great pleasure for me in India, either physically or in seminar virtually. My name is Hesham. I'm a director of Institute of Bioproducts Development, University of Technology, Malaysia. And today, I would like just to share with you my more than 30 years experience in the fungal technology. Whereas back, I have did my PhD, finished my PhD 25 years ago in the field of mycology. And through all those years, I usually consider fungi is a very unique microbes. It's not easy to cultivate in a large scale compared to other unicellular bacteria or yeast. Therefore, one of the main topic, I'm running this research more than almost 25 years, which is attracting me basically as a microbiologist, and after that as a process engineer and industrial biotechnologist, is about the fungal morphology. For my talk today, it's about the fungal morphology in biotechnology industries, taming a wild microbes. How to tame these wild microbes to control morphology for large scale and why we are working for that. Fungal biofactories in industrial biotechnology market as introduction, I will give first. And then understanding growth morphology, following by from spore to fungal macro morphology. And then I give a case study, which is actually not old and new process, which is I developed almost more than 20 years ago, but I added on new informations and the new knowledge in this cultivation process. And then I give some conclusion. Why fungi? This is the question, which is everyone.
Professor Hesham, sorry to interrupt. Are you trying to move the slides? Because we are on the first slide itself, actually, now.
Oh, I'm now in the third slide.
But we can see only the first slide, actually.
Oh, okay. Then I think your advice about go through PDF.
Yes, yes,
Go now
please.
through PDF.
Yes, yes.
Then
Please.
I stop sharing now. Can you see now the slide?
Yes, I can see it, actually.
Okay. Can you see now the second slide?
Yes, 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 we can.
Okay.
Thank you.
As mentioned, we've gone through the lecture content for understanding the fungal biofactory in industrial biotechnology market. Sorry. And then understanding the growth morphology, and from spore to fungal macro morphology, and then go through a case study for glucose oxidase production by recombinant Aspergillus niger, and then we give you some conclusion. First, when you're going to talk about why fungi, this is the question. If you talk about fungal biofactories for production of different metabolites in fermentation industry, There are many types of fungal biofactories, like Acremonium crysogenum, which is the most famous one, which is produce cephalosporins and penicillins, and used in pharmaceutical and manufacturing company is usually it's Novartis until now, and Lupton in India. When you go for Aspergillus awamori, it's a biofactory for many food enzymes like amylase, glucoamylase. They need to be in a filamentous growth and small pellets with a mycelial network. This is based on the literature. And it's the process of nosozymes in Denmark. Then if you go for Aspergillus niger, Aspergillus niger is famous for citric acid. And usually it's recommended to have a billet not exceed 2.2 millimeter diameter based on the literature. And the enzyme like glucose oxidase should not to increase the billet more than 0.4 millimeter diameter. Phytase, pectinase, protease, glucoamylase, and chymosin. For this marvelous strain of Aspergillus niger, like example, they can produce more than 35 products. But you need to switch from one product to other, not only by genetic means, but even for the wild type, by adjusting the pH or cultivation conditions, medium composition, but morphology. As I show here, like example, same microbe, if I need to target to produce glucose oxidase or enzymes, biopellet should not increase more than 0.4 millimeter. But if I need to produce citric acid, this can be up to 2.2 millimeter. If you go for Aspergillus oryzae, it's preferred to be in a filamentous growth. And it's produced lipase, invertase, and protease. And if you go for Aspergillus teres, for itaconic acid, they need hairy and less dense pellet structure. And the pellet not should to increase 0.5 millimeter diameter. Secondary metabolism, in this case, like lovastatin, they have the pellet should not to increase more than 2.5 millimeter diameter. Same if you go for cordyceps, they need to have a filamentous or small pellet, but they have, like example, produce a cordycepine and many bioactive compounds. Fusidium coccineum, which is for fusidic acid, and many other products. You can find here some information related to link between products and morphology, even for the wild type strain. Even for here, like example, Rhizopus oryzae, for lactic acid production, fumaric, the smaller the pellet is much better, between 2 and 2.5 millimeter diameter. Tulipoclidium inflatum, which is produced cyclosporine A, which is immunomodulators and antibiotics compound, need to have smaller pellet, not less than 0.25 millimeter diameter. First, if you would like to say understanding the fungal morphology, if you need to understand the fungal morphology and spore germination, there are two stages. First, from spore induction to spore germination. And the second is from spore to mycelium and biopellet formation or filamentous structure. When you go from mycelium, I need to induce sporulation. Then I'm going to make a mature sporangia. Then I'm going to produce a dormant spore. Then I need to have another induction of spore germination. And those factors induce sporulations are different completely from those induced germination. And then once you have spore swelling, increment on the radial growth, then we have isotropic growth. And then this is for rounded spores. I'm not talking about irregular spores like those in Fusarium and others. This is another model, which is still under study now to have a complete modeling of this one. But I talk here with such spores like Aspergillus groups, Aspergillus niger and Aspergillus teres, Aspergillus oryzae, and all those fungus, they have a rounded or spherical spore shape. Then I am going to have a polarized growth, germ tube germination, and after germination, I'm going to have a polarized growth or germ tube elongation. This is one of the strategy. Then you're going to have a mycelial structure. But there is another model because there are two types of spores. One, coagulative agglomerations, and the other one, non-coagulative agglomerations. And both can make a micropellet or a biopellet formation. In aggregated spore style, like Aspergillus niger, they germinated spores, they further grow together in aggregate. Then they give a compact structure of biopellets. The other dispersed spore structure, like Aspergillus awamori, most of the case, they make the hyphal elements, and those branching together aggregate based on some bioprocess engineering parameters, and then they make hyphal non-coagulative spores or make dispersed mycelium. They have these two options. But as I showed to you, each morphology, growth morphology from the same microbes or the same strain can yield totally different metabolites. If you look for spore germination and formation and germination, spore formation induced by stress, signaling, oxygen, chemicals, and many other factors. Then those cells need to be some stress to have, then to produce sporulations. And some chemicals, some minerals, we know that in the process. If I put some minerals, I induce sporulation straightforward, or some nutrients. We know it's stressing. Oxygen is required for sporulations of those aerobic microbes or those asexual reproductions, spores like in Aspergillus groups. And then we go for isotropic growth. How the cells start dormancy to start germinated. And there is environmental factors, signaling, and chemicals, and many other things which is can controlling or governing this process. Why germ tube formation, polar growth? Then I'm going after increment of the radial growth. I'm going to find the germ tube. What is this germ tube? Increased growth rate. Like you find in a bowl, you're going to increase pressure. In some parts of the wall, which is weaker part, going to make elongation. This is exactly, if you're talking about kinetic of the formation of the germ tubes. It's increased growth rate and enzyme excretion and signaling. Then this is making a germ tube. But from spore swelling to germ tube formation, as I mentioned, not all factors depend on all microbes. Maybe low temperature increase swelling, maybe high temperature increase swelling. It's depend on the type of microbes and type of spore. Light is inducer, the pH, signaling molecules, inorganic nutrients, organic nutrients. And AW is one of the main factors here. And they have some signaling molecules, and they're going to have a germ tube formation. People say in some research, not only for swelling. Okay, AW is very important for penetration of water inside the cells or inside the spore structure. And then not only that, they have sometimes enhance or making some signaling for germ tube formation with more water entering, which is the AW factor. Fungal morphology, riding wild horse, exactly like how to control this wild horse. Bioprocess platform for recombinant protein production by Aspergillus niger, which is going to present, showing you a process, a real process, which is I developed long time ago and improved in between now in many directions for scaling up of the process and keeping the same morphology. First, why glucose oxidase? What's the main target of the platform design? Increase the production and excretion of glucose oxidase using the recombinant microbes. and then study the relationship between fungal morphology, the shape of fungal growth, and enzyme production excretion in stirred tank reactors. Then high cell density cultivation of Aspergillus niger in a stirred tank bioreactor, and scaling up of the production process to pilot plant level up to 200 liter. Now we scale up the process up to 1,500 liter. Why glucose oxidase? Actually, being as a, now you can say, bioprocess engineer with a microbiology background and chemistry, I like usually to understand the big picture of product. Then I understand, is this going to be in high quality form, it means pure form, or bulk form, food grade, pharma grade, or what? Glucose oxidase can be used in analytical chemistry, in diagnostic and biosensor, in the food industry for fructose production, gluconic acid production, and used as preservative, natural preservative, which is used in egg industry. But the main three things usually you're looking for: price, purity of the final product, and CGMP requirements. Is this need CGMP or not in this production process? For this process, once you are not producing medical compounds, you don't need the CGMP requirements, but you need HACCP requirements or other guidelines for sanitary production, which is make it easy in this process. But first, we need to understand the process bottlenecks. The prior process bottlenecks. If you see, like example, what factors affect the production, we can come up with 200 factors. But what's the bottlenecks, which is the main factors? If I can control, I can reach 90% of the theoretical productivity. For microorganism, you know, there are two types of microbe, Aspergillus niger and Penicillium amaga-cantiense. Those both produce intracellular enzyme. What does it mean intracellular? I need to break the cells. Extra step to break the cells after fermentation. It's not about one step after that. But once I open the box, the cells, I have everything's inside. If I need to purify glucose oxidase among this very big pool, it will be difficult. Then the second bottleneck is a morphology. As I mentioned, in submerged culture, is they produce pellet or filamentous? This is the big question. Substrate, most of the people use glucose as a carbon source. Once I produce glucose oxidase, it converts glucose to gluconic acid, straightforward. It means I don't have substrate anymore. The first problems have been solved using genetic engineering approach to having Aspergillus niger with the capacity to excrete the enzyme outside. This based on NRRL3 Aspergillus niger. We have donated the name OD3-18. And these microbes, they have GBDA promoter from Aspergillus niger and alpha amylase signal sequence. Alpha amylase is usually excreted out of the cells. Once the cells have alpha amylase signal sequence, peptide sequence, then I can excrete the compound with the structured gene of glucose oxidase and N-terminus of glucose oxidase. Then the first problem for having intracellular product have been solved. Then I have extracellular products through these techniques. This was in collaboration with the Technical University of Darmstadt at that time, Prof. Jung and Rutovetsky and Prof. Gassen. Then, as I think everyone knows about this slide, you need just to optimize production medium, different nitrogen source, different glucose concentration, different phosphate source, and different yeast extract concentration. And we see that yeast extract is one of the main factors affecting the yield. If you see here, the yield of glucose oxidase excreted, this as extracellular products, which is about 20 microcatal per liter, which is the early experiment we have when we use just 1 gram per liter, when just produce 1 gram per liter of yeast extract. Therefore, we keep between 1 and 2 grams per liter, but we calculate here the number of pellet and the pellet density in milligram per millimeter. We find here the pellet density decrease once we increase the yeast extract concentration, and the pellet diameter increase in terms of millimeter. Furthermore, we investigate the growth or the fungal biopellet formation in a shake flask. When you go in a shake flask, we have seen that the biopellet is not homogeneous in structure. We have a different layers, different layer A, which is exposed to oxygen, which is a dense layer because this microbe is aerobic microbe. The layer B is less dense because it's not exposed to oxygen. Then I have another dense layer, which is just a germinating spores. And in the core, I have a layer which is of spores on the core of the pellet, some even non-grown spores. Why they cannot grow? Because not exposed to oxygen. As I mentioned to you, to break dormancy here, I need to have some factors. And those in the center of the biopellet, where is the limitation of oxygen or KLa value, means they cannot germinate easily. Then we go cultivation in 5-liter stirred tank reactors. When you see here cultivation in 5-liter stirred tank reactors, we see calculate for extracellular and intracellular product. And this is the total of glucose oxidase, which are reaching about 100. In our previous cultivation in a shake flask, we have extracellular products of 20 microcatal, but here we can reach about 80 microcatal. The same time, you can see here for glucose oxidase, this is the extracellular value. Sorry. For extracellular value, can reaching 80 at the end of cultivations, but almost 55% still have intracellular, and we have extracellular products. It's a mix between both extracellular and intracellular product. And this is the biomass. We have the biomass of 4 grams per liter. You see here glucose consumption go fast. At the same time, gluconic acid go also as the function of consumption of glucose. It's not used mainly for growth because I use 80 grams per liter in this media. Not the 80 grams per liter not going to produce 4 grams biomass, but it's mainly producing gluconic acid. Then we follow here biopellet. What we conclude here, substrate limitation and gluconic acid formation. This is another problem here. And the biopellet is a critical because the biopellet diameter is about 1.5 millimeter. Then we have in this 5-liter schematic programming system to see how we cultivate. We control the pH, dissolved oxygen temperature. We look also for the outgas analysis to understand the CO2 production and oxygen consumption, and all those together under the Opicon control system. And we have sodium hydroxide to adjust the pH because I produce gluconic acid in this process. Interestingly, here in the 5-liter reactors, we can produce filamentous or pellets under the same cultivation condition. But we see here another critical factors. With the same agitation, same media, same aeration, same everything, I can produce filamentous or pellet. What we have seen here, we usually use spores from cell bank. And we just make one cultivation on a plate. We collect spores and use to inoculate. I'm usually for aspergillus, even for industry, they can still use spores as inoculum even for big fermenters because they worry about the morphology. Once they start with the spore, they can control. But in this case, we have 2 processes. One, from cell banking glycerol culture, I just make straightforward one plate using CM medium and inoculate the bioreactor straightforward. The other cultivation is we subculturing. It means the spores become more hydrophobic. In this case, it's make more filamentous structure. And if you see here, just change from filamentous to biopellet, the complete kinetic of the cultivation have been changed. And this is proven too much how significant is the biopellet formations in this process. I can see here dissolved oxygen value. You see, drop is too much. It means the cells are very active compared to this one. Sorry. Compared to this one. Compare this one compared to this one. This is more deep. Dissolved oxygen here reaching almost 20% saturation. This not go off 60%. It indicates that this is less active, physiologically less active compared to the filamentous. And production here was also totally significant differences. In case of filamentous, I can reach about 250 microcatal, like here, but in the pellet formations, it's the production here, which is very less, about 100 microcat. When we study here, these two cultivations in more depth, to have the carbon balance and look for dissolved oxygen and CO2 production, oxygen production, we see that filamentous produce more carbon dioxide and consume more oxygen compared to biopellet or pellet formation. To solve the problem of gluconate production and limitation of substrate, we look for different carbon sources. And we figure out in this type of microbes, these recombinant microbes, we can produce xylose, mannose can produce glucose oxidase, better than glucose. But because of the cost, mannose is very expensive compared to glucose, but xylose with reasonable price. We make its cultivations using xylose. But we compare xylose, fructose, which is can also induce the production, and glucose. If you see here, different cultivations, in the left side, in figure A, you can see that the maximal production using xylose, followed by fructose, followed by glucose, and then fructose. The same time of a biomass production, the maximum is xylose. When we study the excretion, because the morphology are different here, even I just changed the carbon source, fructose making the strong biopellet, which is limit the production of glucose oxidase. I see xylose, better excretion, almost like glucose in the culture media, better than fructose, because of intracellular, extracellular reaching about 50%, 50-50 here for the fructose after about 120 hours of cultivation. We go in more depth about fungal morphology in this process. We study the micromorphology and macromorphology. The micromorphology about the spore diameters distribution, spore germination, hyphal length, and hyphal branching. And macromorphology, we go through the biopellet distributions and biopellet diameters and pellet structure. From literature, macromorphology can affected by different factors, which is strain-dependent factors, medium composition, and cultivation condition, which is called bioprocess-dependent factors. But which factor I can work with? It's a big dilemma here. All those in the literatures can affect the micro and macromorphology. But in scaling up in a process, we usually look for something which is we can control in scaling, which is the aeration and agitation rate and the type of the bioreactors. Then I use a fluorescence microscopy, very simple fluorescence microscope with image analysis system, and make a monitor and look for the biopellet structure. We follow growth micromorphology, how the spores germinated, how they swallow down. Here, like example, we find after 0 hour, 1 hour, 2 hours, 3 hours, because of not all diameter of spores are equal. Spore diameters are variables, by the way. Is this in the top of the conidia or in the bottom of the conidia? Therefore, but when you have spores, diameters are different a little bit in terms of micrometer diameter. And we have some distribution, even though it's very little, you can say between 2.4 to 4.8 micrometer, but we like too much to understand these differences in distribution of the parameters, of the diameters. And then we follow up this of swelling of the spores and also germ tube formation. And then we see from swelling to germ tube, we find the primary parameter, germ tube germinations, as a function of shear stress. At different agitation, we can follow up how it's go up. We see that germ tube length is proportional to the agitation in the big bioreactor. It means they make a faster growth rate. The same for probability to have two germ tubes is giving the same. And the germ tube volume is also increased as well. It means I figure out here 800 RPM agitation affecting the kinetic of germination, swelling, and germ tube formation. But what about the production? We find the best production at 500 RPM. At 500 RPM, you can find, sorry, it is some of the slides is in German because I was delivering some lectures recently in Germany. And the UPM is Umdrehung pro Minute, which is RPM, round per minute. BTM is Biotrockenmasse, which is the cell dry weight. And they just, sorry, it's for that. I didn't translate it again to English. And, but I think it's clear for you. We see that 500 RPM is the best for total glucose oxidase production, but the maximal biomass is at 200 RPM. We look for the macromorphology, how it's work. For the billet diameter, for sure, there is a standard deviation in the diameter. If you see here, for 800 RPM, standard deviation is very low compared to 200 RPM. But we try to find a relation between production and macromorphology. Therefore, we make a carbon balance of different agitation speed. We figure out that a maximal CO2 production is 500 RPM. Is this the most physiologically active cultivation? We've proven that through morphology, and we follow morphology using image analysis system with acridine orange staining. Why acridine orange? Acridine orange staining can differentiate between DNA and RNA. Considering that the RNA reach, it means enzyme reach. The DNA reach is more dormant, not producing enzymes. Then we calculate, it was here very obvious, you see here under microscope, this outer layer is the productive layer. The inner layer is not producing enzymes. Here is the mix in 500 RPM. Here is orange, almost like a mix, and they mostly are productive. And we calculate this diameter once we know standard deviation. It was very tedious calculations here because I cannot take one sample, but I need to have at least 100 pellets or micro pellet to see the distribution, and we calculate the outer layer. And then we considering the volume active is equal to the total minus inactive part. Inactive part is the green part. And we see the active fraction of the biopellet. When we calculate the active fractions, we recalculate the biomass. We see here, this is the total biomass, 30 grams per liter. When I just calculate the outer layer, it's about 8 grams per liter. And this percent of the active biomass is about 30%. It means 70% of my biomass is not producing enzymes. When I calculate the yield coefficient, microcatal per gram, considering 800 is totally all is active. And 200 RPM, I have these two layers. It means 30% only are active. When you look for the yield coefficient, this is the yield coefficient when I take the total biomass of 200 RPM. And this, when I consider the active biomass, it's giving the same value of 800. It means theoretically I'm correct and practically I'm correct. So it means here we have active and inactive biomass. Only the outer layer of the biopellet is the productive biomass proven by different cultivations at different RBM. Then morphology here is the main limiting factor of the enzyme production and excretion. The pellet size diameter should not exceed 400 micrometer. Then we go through high cell density cultivation platform to make higher biomass under the desired morphology, and we can reach up to 1,500 microcatal. You remember we were starting with 20 microcatal in this process, and then it's significant improvement. And boundaries of bioprocess scaling up in a bioprocess engineering. Usually when we scale up a process, we look for aeration agitation, and we need to work within this hexagonal structure. If I make more aeration, sorry, I just need to, aeration agitation here. If I make more aeration and more agitation, it's the cost of the process very high. Moderate agitation and high aeration, there will be a foaming. If I have high aeration, low agitation, there will be poor mixing because of the diameter of the oxygen bubbles, it will be big. If I make moderate aeration and low agitation, I will going to have oxygen limitation. Low aeration and moderate agitation, CO2 inhibition. Then I work within a certain limit. Then when we scale up a process, we look for specific power input B over V, or impeller tip velocity, or theta 90, volumetric mass transfer, KLA, or Reynolds number. But when we scale bioprocess of fungus, we consider these, but the most important one is a morphology. If I scale up of the process and I change morphology, then it will become the bottleneck of mass filamentous microorganism cultivation process. In summary, we start in the shake flask 20 microcatal, 100 microcatal when we move to 2-liter bioreactors, 250, then we go up to 1,800, which is, we can say, 90-time increment in the process when you make feed batch cultivation using a xylose and high cell density cultivation strategy. Then we develop the know-how, but this know-how is based on growth morphology. If there is any change in the morphology, which should not to be, then the yield will be different. With this, I would like to say thank you very much from Institute of Bioproducts Development. We have these two big buildings, with this our laboratory and pilot plant. This is the administration building, where I'm sitting here now talking with you, and would like to say thank you very much for all of you for attending this lecture. Thank you.
Rohit, are you there?
Yes.
Thank you very much, Professor Hesham. Thank you very much for that wonderful talk. Maybe you can stop presenting your slide so that we can have a better visual of us, actually. Yeah.
Stop presenting. Yes.
Yeah.
Okay.
Yeah. Maybe now we can go. Maybe we can go ahead with the question and answer session as we planned. Maybe we can spend next 20 minutes having some interesting discussion. Maybe Dr. Mohan Kumar can ask his first question. Dr. Mohan Kumar, please.
So it was really a great experiment what you have conducted for upstream bioprocessing. So some of the downstream bioprocessing strategies, what have you planned for your product, and can it be influencing on the product yield and product productivity? And coming to your upstream processing, have you tried with an oxygen role of oxygen in the production and increase of yield by making the an aeration adjustment in the aeration or something like that?
Yeah, thank you very much, Dr. Mohan. It's very critical questions you mentioned, which is give a complete picture about the process. Downstream process in this case was easy because of we excreted as extracellular product. Then with ammonium sulfate precipitation, we just precipitate my protein. Further purification is required if I need for analytical grade. But with food grade, it was more than enough. It's just very simple process for precipitation of the protein. In terms of oxygen limitation in this process, I like to match your question. Usually when we design a process for scaling up, we try to avoid cost. It means if my dissolved oxygen above 20% saturation, I don't go through any oxygen supplementation. I go through oxygen supplementation only under high cell density, where I cannot achieve 20% saturation. Even I reach sometimes oxygen limitation. But one of the main issues, we work with the oxygen here for scaling up. We try to avoid, again, any oxygen because of two factors. Safety in the pilot, in the plant, because once you have oxygen, pure oxygen, you need to have oxygen generator. It will not have a cylinder. And this is one, according to Hesham, one of the risky things. The second thing is the cost. How to increase dissolved oxygen in a reactor? We can do it through bioprocess engineering parameters. With changing of the impeller shape, changing of the impeller diameter, I can increase Reynolds number and oxygenation as well. Oxygenation here in the bioreactors, I just need to have the smallest bubbles as much as I can, small micro bubbles. Even sometimes membrane aeration is good. The game here, how to make small bubbles, because the total surface of the bubbles indicating the oxygenation area inside the reactor. But the last choice, usually we go through oxygen if we fail in all bioprocess engineering parameters.
So some of the slides showed that the xylose and mannose has increased the yield, but the glucose oxidase is not specific to the substrate.
No, for this strain, recombinant strain was not specific for the substrate.
Thank you, sir.
Thank you.
Thank you. I request the audience not to just speak without without me allowing you, because otherwise it will be chaos in this platform. Thank you. An audience named SPJS1 India. May I know your name and what?
Yeah. Hi, Shinoy. This is Dr. Senthil.
Yeah, something. Yeah.
Sorry, sir, very nice presentation, because it's a bioprocess related. In the world, it's a very unique. The bioprocess research area itself very unique research area. And we are also working on filamentous-based fermentation and production of enzymes, like in the inulinase. Recently, we have interacted with amino enzymes from Japan. They're also interested in this type of food enzymes. Sir, my question to you is on your glucose oxidase enzyme production through xylose feeding and through fed batch, right, sir? Suppose if you compare same thing with a recombinant based glucose oxidase enzyme production in East Picha Pasteuris system extracellular, whether that is showing the higher yield because it is a short fermentation time, same fed batch, 1000 cell OD per ml. Here also, like it is long-term fermentation, whether in cost economics wise, recombinant based glucose oxidase is better compared to the filamentous based long-term fermentation. So just I would like to know, sir, cost economics.
Okay, thank you. Thank you very much, doctor. It's very, very tricky questions. I like it. Picha Pasteuris, I'm familiar with this high cell density cultivation using OX1, OX2, which is the new process. But considering something now, homologous expression and heterologous expression.
Yeah.
In terms of regulations. What I did here in this platform is homologous expression, not heterologous.
Okay, homologous expression.
Yes, because I didn't choose the biofactory. I didn't change the microbes. I just make alpha amylase signal sequence and N-terminus. That's it. In this case, your regulatory bodies not going through you to say you are biosafety level 2. This is the tricky things here. Then you are still considered as a natural product. You are not doing it genetically modified. And this is part of the selling point. The second thing, big capacitors, I agree with you, it's fast growth and so on. But I don't know about the yield here because it will be heterologous expression system.
Yeah. Most of the diagnostic industry to produce that glucose oxidase used in the biosensor is through E. coli, intracellular glucosidase enzyme. So, like in terms of cost economics, that's a one example only. But fungal mycelium-based things has been considered as a cell factories by many products like citric acid, taxols, many other products also. But it's a good insight, sir. You have shown a good fermentation work. Like we will also follow the similar type of fed batch fermentation, especially this xylose. Xylose is a wood sugar. It acts like an induction. Thank you, sir.
Also, I would like just to mention to you something. The process was not stopped as I showed to you today. We did also immobilize cell system. Then the production time was just 24 hours.
Oh, that's wonderful. Fantastic. Great industry-friendly process.
Okay, yes.
Thank you, sir.
Thank you, Senthil. Professor Hesham, I would like to add a couple of sentences about Professor Dr. Senthil. Actually, Dr. Senthil works for the CSIR, Institute of Microbial Technology, which is in the northern part of India. So they have a wonderful facility for fermentation, actually. I will give more details later. Thank you, Senthil, for joining and asking some wonderful questions.
Thank you, Shana.
Yeah, thank you.
And Doctor, please, I will write down my email.
Yes.
I love you much to visit you and visit your facility in my next time I go to India.
Yeah.
Sure, sir. Sure, sir. Welcome, sir. Yes.
Thank you. Dr. Rohit Sharma, please.
Yes. What do you think, on the just now for the recombinant thing or the wild strain? If one wants to go for the kind of development of any technology kind of thing, so there is always a question on the whether going for the recombinant thing or like going for a biodiversity thing and target based sampling and going for a wild strain. So what are your views on that?
Thank you very much, Professor. I tell you what, me personally, I love wild type. We go to recombinant unless there is a must. And I go for homologous first, not heterologous. Okay. If I can solve the problem of production and economy of the process using morphological parameters, physiological parameters, bioprocess engineering parameters without touching the genes, I love to do it. If I need to touch the gene, like in this process, just to make the cells excrete outside the cells, then I'm still homologous. I'm still use the same biofactory. I know some process, like example, insulin, you don't have choice. We don't have any microbe who can produce naturally insulin or those human proteins. Then there is a must to go for recombinant. But those which is constituted can produce by microbes, I love to continue, be calm, be patient with the microbes step by step until we reach a target together.
Yes. Thank you, sir. Thank you very so much. Yeah.
Mr. Anil Behera, please.
Hi, sir. It was a nice talk, sir. I have a basic question. I didn't understand about the isotropic growth of fungi.
I didn't catch up.
Isotropic growth of fungi.
I think kindly be louder, Anil. You are not audible.
Sir, about the isotropic growth of fungi.
Isotropic.
Yes, sir.
Let me interrupt here. I think Anil Behera plus first of all mute yourself. There is some noise on your side. I think Mr. Anil Behera wanted to know about the isotropic growth of fungi. So he was trying to get more information about what do you think? I mean, what are your how can he get more information about the isotopic growth of fungi actually?
Yeah, I mean, isotropic is one of the phenomena that's for the fungal growth. And isotropic, this is one of the main things also we consider during scaling up of the process and industrialization. But I mean, there is no generic answer for that because of it's specific for each microbe.
Okay, I think we can catch up with that, and he can contact and try to get more information.
Yes, sure.
Thank you, Mr. Anil Behra. Amy Jose, please.
Doctor, am I audible?
Yes, you are audible. Yeah.
Doctor, am I audible? Doctor, am I
Yes.
audible?
You are audible. Yeah, please ask your question. Thank you.
Doctor, I would like to know what's the mechanism of glucose oxidase in the preservation of food? Like how it is preserved the food?
How they preserve the food?
Glucose oxidase.
Yes, because they produce hydrogen peroxide.
Sorry, sir?
Because when they produce glucose oxidase plus glucose, they produce gluconic acid plus hydrogen peroxide.
Okay. Is it very apple cider vinegar? No, apple cider vinegar. Apple cider vinegar. That is also produce some acid. No, it is also preservative. It is very less cost. Apple cider vinegar.
Okay, wait. Let me just, Mr. Jose, can you speak slowly and clearly? Maybe the speaker is not able to catch up with your words. Please, thank you.
Okay. So, okay, sir. So, what are the which food you usually use glucose oxidase to preserve? What are the foods?
Okay, I'm not use glucose oxidase as a general preservative. There are many cheaper preservatives.
Okay.
Glucose oxidase is used for dry egg preservative. This is the biggest market for them. When they produce dry egg, it means which is in the egg market, they use this as preservative because of not affecting the aroma.
Okay. Sir, dry gas does not have any water content. So, what is the use of this using preservative? There is no dry. The dry egg is very dry. No, there is no water content. So it is not. It's already naturally preserved. So what is the use of using this glucose oxidase?
They mix together during the process of drying.
Okay. So okay. Thank you, sir.
Thank you.
Thank you, Mr. Joseph, for your nice questions. I think last question from Risha Pimenta.
Hello, sir. Great session. My question was you spoke that upon subculture, spores get hydrophobic. Now, are there any chemical treatments that would avoid that, like by reducing the surface tension and then reducing the hydrophobicity?
Yeah, thank you very much. This is one of the tricky industrial questions, but I will answer you. Okay. Sometimes we use twin 80 in a concentration of 0.1% to play with the hydrophobicity and aggregation of spores, which is makes it easy for collections. You are not just collecting with a saline solution, but if you add very low concentration of twin 80, then which is decrease hydrophobicity and make it easier for collection and dispersion of spores. Same for if you're going to have multi-stage inoculum, we usually recommend to use very low concentration of twin, but before the main vessel. I'm not using the production vessel twins, just but in very low concentration, 0.1% or something like this. It means 1 gram per liter.
Okay, sir. Thank you. Have a great day.
Thank you.
I just said the last question, but someone has raised his hand. Maybe I should allow him to ask question. Dr. Chandan, please ask your question.
Yes, sir. Actually, sir, this is very simple question. I have gone through all the slide you have discussed. In that all discussion throughout your lecture, you have used the term biofilms instead of saying
Okay, Dr. Chandan, you some audio issue there. You try to fix it and come back to me, or maybe just type in the chat box. Thank you.
Okay.
Okay. Just send an email to Professor Hesham. Probably he'll be happy to answer your questions.
Yeah, okay.
Dr. Rohit, over to Dr. Rohit Sharma, please.
Yeah, thank you, Dr. Shnoy. Am I audible? Yeah. Thank you, Dr. Hesham, for a very nice talk and focusing on the scale-up things. I think while working on the applied and industrial biotechnology kind of thing, so two major things like development of technology at the lab scale, and then the important thing comes up to is the field trials or the scale-up thing. And I think it's a very different ball game altogether when we are doing a scale-up thing or a fermentation thing, as compared to the handling of fungal culture for their physiology and biochemistry. So I think thank you so much, Dr. Hesham, for the very nice talk, and I hope that we'll have some kind of association with respect to the Association of Fungal Biologists and Micro India in future, maybe kind of things. Thank you so much. Thank you so much.
Yeah, thank you very
Dr.
much.
Deshmukh.
And actually, I'm yes, Prof. Dr. Sorry.
Actually, I'm thankful to you for giving a nice presentation and accepting our invitation.
You are more than welcome, Prof. And I hope this will strengthen our collaboration with the Indian Society and the Mycological Society for further collaboration in research and maybe for products developments for the market, which is I like usually to do with many industry. Thank you.
Yeah, sure.
Dr. Rohit, let me take this opportunity to propose the vote of thanks now, maybe. Yeah. First of all, Professor Hesham, thank you very much for spending your Sunday with us and giving a wonderful lecture and patiently answering the queries the audience had. Actually, thank you very much for that. I hope this association, this interaction will continue. We would like to have you with us for Micro Asia and Micro India in a different role. I would contact you about that in a couple of days. Thank you very much once again for giving this wonderful talk. And secondly, I would like to thank the Association of Fungal Biologists for collaborating with Micro India, Journal of Indian Fungi for this lecture series. Thank you very much, Dr. Deshmukh, Dr. Shashireka, Dr. Gosavi, and some wonderful people. And then I would like to thank Dr. Rohit Sharma, the managing editor of Micro India, for his excellent support and excellent help. He's Mr. Dependable of Micro India and Micro Asia. Whenever I need him, he will be available and he will do the job for us wonderfully. Actually, thank you, and a wonderful audience today for your active participation, asking some very pertinent questions. And you are our strength, as I always say, you are our strength. We are here to serve you. So that's where we get our motivation. Thank you very much, ladies and gentlemen. With this, we sign off. Feedback form will be link to the feedback form will be shared in the chat box. You're welcome to submit your feedback in next 10 minutes. Thank you, Professor Hesham. Have a good day there. Dr. Deshmukh, have a good day.
Yeah, bye.
Dr. Rohit, bye-bye. Everyone, bye-bye.
Thank you.
Bye.